All right, so our very first speaker is Monica Paez-Vacas. Monica, go ahead and take it away, please.
Okay, so can you see my slides and can you hear me?
You look and sound great.
Okay, thank you. Welcome for this last session of the meeting. I'm usually working with frogs in the tropical Andes of Ecuador, but today I'm going to talk about a project I did during a postdoctoral fellowship, 2 years in the Argentinian Patagonia with lizards. Wait. Okay, so understanding what are the processes that originate, that create the biodiversity and let it persist is an active area of research and remains a relatively unexplored frontier in the southernmost regions of South America. There are several mechanisms that are really interesting in explaining these processes, and hybridization is one of those, and now it is recognized as a widespread phenomenon across many taxa and a key process shaping variation and even adaptation. So here I have put some of the very famous examples about this. Hybridization also offers valuable insights into the mechanisms involved in population differentiation and reproductive isolation, and ultimately speciation, phenotypic change over time and space, etc. And if you're like me and you don't know the Lialemus lizards that I just met when I went to Argentina to work on them, and it was a fascinating experience, let me introduce you to the Lialemus lizards. This is a very rich group that is distributed from the southern parts of Peru to the southernmost tip of the South America. And it has more than 290 species, making it the second most diverse amniote genus globally. It's 25 million years old, and they live in very extreme environments like the Atacama Desert or the Patagonian steppe. And some species can even found on snow, salt pans, and there's one species in Peru that can be found at above 5,000 meters in elevation. They've also have gone several times have gone through the transition between oviparity and viviparity, and also the opposite, viviparity oviparity. There's one parthenogenetic species. There are some species that have also transitioned to a herbivorous diet, but without compromising some physiological traits. So it's a really, really interesting group, and that has many people trying to understand why are they so diverse. If we see this previous work on the speciation above and the extinction rates below of the family Liolemidae, we can see that Liolemus, this very rich genus, doesn't have a very high speciation rate compared to, especially to other genus within this family, but the extinction rate apparently is rather low. And something that I also found really interesting about this group is that they have this generalist morphology, and that they live in these heterogeneous and fluctuating environmental conditions in southern South America. So some experts think that perhaps this lack of specialization may facilitate survival by enabling niche shifts under environmental change at different evolutionary scales. Hybridization is very common in this genus. I'm just putting here two examples showing that it can occur in groups that have diverged recently, but also in other groups that have diverged before. And hybrid zones are mostly found in south central Patagonia, and there is this range, this area, this latitudinal area, where there is richness hotspot, and the speciation rate is relatively high, and the extinction rates are relatively low compared to other species in the same genus. Within this area, there is one group, the Liolemus spitzengeri group, that you can see here. There's this distribution. This is the phylogeny from the mitogenome. And here I'm going to talk about this group in the bottom part of the tree that we can see. One group that I call it Melanops chewen, or northern Xanthoviridis in orange, which is the central one, and Pizzingeti in green, the southern one. And one thing that I want to mention here is that this group is so diverse that most of the work, not all, but much of the work in the last decades has been researchers trying to figure it out, the phylogenetic relationships, how many species they are. So we're starting to understand these evolutionary relationships, and I think a very exciting time to understand some of the processes are coming ahead, but there is a lot of work to do as in other systems, of course, in the region. So previous work in this group has a sample latitudinal transect here and studied if there was hybridization among these species, and they found that indeed there was hybridization. And this is when I come to the story. I go to Argentina to study how hybridization influence phenotypic and genotypic or genomic differentiation in this group across the landscape. So for that, I take a genotype, phenotype, environment approach in which I took advantage of specimens housed at the herpetology collection in the National Patagonian Center in Argentina, and also the DD-RAD loci that was sequenced by Grammer's study. I obtained more than 1,500 loci, and I want to show you some of the things I've been finding so far. So for the phenotypic variation, I measure 15 morphometric data, usually measuring these groups, but I didn't find much variation between populations, although there is a tendency for a larger size, this is body size, to the south. And this is really similar in other measurements that I took. If I show you phenotypic or morphometrics in a multivariate space, you can see that there is not differentiation in the groups, and especially if I remove the effect of size. Whereas for the genetic variation, we can see that the northern populations are really grouped together, and then as we move south, we can see this clinal kind of variation here with this DD-RAD SNPs that I studied, and you can see how this variation looks like in the geographic space. If we see the graphs of each individual and each site, we can see that there is definitely some hybridization there. And I also wanted to know what are the patterns of putatively adaptive genetic variation. So I use PCAdapt to find loci that had variation that differ from the background, the rest of the loci, and I obtained 24 outlier loci. And it's interesting that if you compare this variation with the all loci variation, we can see that the variation within populations increases, and the differentiation among or between populations decreases. And this is really interesting because it could be that some of those loci or loci linked to these ones are under some convergent selection because they share similar environments, or maybe it could be some mechanism like balancing selection that might be maintaining polymorphisms. And I wanted to check this second hypothesis, so I wanted to know if there were some signal of balancing selection. And for that, and because I'm using DD-RAD, I calculated the GMSD with the purest populations I could find. And here, if you have positive values of this GMSD, that could be a sign of balancing selection and or sudden population contraction. And what I found in the three groups or species is that there are many loci with positive values, and there are some that have values larger than 2. So there might be some signal, and I need to explore these further. I also wanted to know if there was evidence of gene flow or reproductive isolation along lines. And this is a work that our teammate did. Kevin. And so here you can see the clinal variation, the hybrid index in the northern and in central cline. And this gradual transition suggests that there has been secondary contact and there is introgression and gene flow across the contact zone. Interestingly, this gap that you see there, if we see the map, is not really related to genetic distance. When we look at the southern cline, central and southern groups, we see this gap, a limited hybridization apparently between these central and southern populations. And again, this gap is not related to genetic distance. So I really wanted to understand what were the environmental and spatial processes that drive this genetic variation. And here you can see how the habitat of this species looks like. And this is like very vast territory that has these characteristics. And I wanted to know how this affected these patterns of genetic variation. So for that, I did regression between genetic data and environmental geographic data. Here you can see two that have some signal, and the stronger and significant was temperature seasonality, and at some extent, at a lesser extent, geographic distance. If we see how temperature seasonality varies, we can see that it's higher in the northern parts of the range. I repeated the analysis separating the two clines, and I found very similar results for the northern central one, but nothing significant for the southernmost cline. And I also wanted to know if there were genetic bases of local adaptation to the environment. So for that, I run GEA, two analyses, and these I need to explore further, but so far I'm seeing that there are some loci that might be related to environmental conditions. And with that, we can see, to conclude, we can see that there was little phenotypic differentiation, at least in size and morphometric data. Fluctuating environment influences the patterns of genetic variation, also some IBD. And there are mechanisms that maintain genetic variation in this group, perhaps balancing selection and also hybridization. However, these mechanisms might not be the same in the studied clines because we saw varying levels of reproductive isolation. There might be differences in times in secondary contact, and there are differences in the presence, absence of ecological barriers or selection. So what's next? Yes, I'm working on the coloration pattern. So far I'm doing some preliminary analysis. I also need to check if there are some loci that overlap between these three analyses that I presented here. And I need to do the genotype-phenotype associations later. Something really exciting is that my postdoc co-director is starting to sequence Liolemos genomes. So I think that a lot of questions we're going to be able to answer. So some exciting times are coming. With that, I want to thank the institution that facilitated that collection before I went to Argentina, my team and my directors in Argentina, CONICET, of course, the Liolemos lizards that as researchers in Argentina, as CONICET, or researchers abroad, thrive and succeed even in hostile environments and really win by the way. So with that, I want to thank you, and I'll take any questions if you have some. Thank you
so much, Monica. All right. So our next speaker is Lauren Bartel. Lauren, go ahead and take it away.
All right. Well, hello, everyone. Thank you so much for being here. My name is Lauren Bartel. I'm a senior about to graduate in 6 days from Harvard University, working with Professor Scott Edwards in the Organismic and Evolutionary Biology Department. Today, I'm going to give you the first direct estimate of the de novo structural variant mutation rate in any wild bird found via long-read sequencing. And the species that we're going to be talking about is the Florida scrub jay. So, how fast do structural variants arise in a wild bird? We know this for humans, we know this for mice, for chimps, for dozens of other model organisms whose pedigrees have been sequenced. But for any wild non-model species of bird, we have no direct estimate. Not one has been published in the literature yet, based on long-read sequencing. And that's not because birds are boring. It's because the technology you need, the long-read trio sequencing, only recently became available enough to point at a wild population. So uh, we have this taxonomic gap. So to make this gap in research clear, in 2013, Bergeron et al. Published a Nature paper assembling direct trio-based single nucleotide mutation rates across 68 vertebrate species. And that's been a really foundational point of understanding comparative mutation rates. But that work and almost everything that has been built on it stops at single nucleotide variants because of this technological barrier that we've had. Structural variants, which are insertions, deletions, rearrangements, etc., of 50 base pairs or more, have been left out almost entirely due to the unavailability of long-read sequencing until recently. So why does this matter? Why do structural variants matter and why were they missed? So a single structural variant can disrupt, duplicate, or rearrange thousands of base pairs in one event. The orders of magnitude more the orders of magnitude more sequence in a mutation point at a single loci with outsized power to change gene function, dosage, and regulation. And short reads physically can't span a structure variant breakpoint because uh, the short reads can often chop them up, and so it's harder to uh, get these structural variants that could be hundreds or thousands of base pairs long. And they can't capture inserted sequence that isn't already in the reference. But now the technology is caught up with PacBio Hi-Fi reads. There are 10 to 25 kilobases at over 99.9% accuracy. So if you pair that with haplotype resolved parental assemblies and rigorous de novo SV detection, it's finally possible to detect SVs in non-model organisms. What was missing was a species with a pedigree to use it, and that's where the Florida scrub jays come in. So the Florida scrub jay was used as the focus wild species for this study. It is a cooperatively breeding passerin endemic to the fire-dependent scrub of Central Florida. It is the only bird species endemic to the state. It's federally threatened and it's declined sharply over the last century due to habitat loss. The population at Archibald Center has been monitored continuously since 1969, with every bird banded, every nest tracked, so it's one of the most complete vertebrate pedigrees anywhere. It's threatened, so mutation rate has direct conservation relevance, and it has a small effective population size with documented genomic erosion despite a stable census, which makes its mutation rate genuinely worth knowing. So before we go into the data that I collected, this neutral theory hands us a prediction. So under neutrality, diversity pi equals for any mu. Now, if structural variants and SNPs see roughly the same effective population size, and they should because they sample the same population, then when you take the ratio of their diversities, the any terms cancel, and so what's left is the ratio of their mutation rates. So I can directly measure the diversity ratio. I can predict the mutation rate ratio according to this theory. In the Edwards lab, Edwards et al. In their 2025 science paper measured a structural to single nucleotide polymorphism diversity ratio of around 0.022 in this species. And from the previous Bergeron data, we know that birds carry roughly 50 to 80 de novo single nucleotide polymorphisms per generation. So multiply those together and we get this prediction of about 1 to 2 de novo structural variants per offspring per generation. So that was the theory that we were going into with this study. So the approach that I used was having 5 parent-offspring trios with 13 birds in total. So we had 1 or 2 trios that had the same parents but different offspring, so siblings. All these were from the Archibald pedigree and were sequenced at Harvard's Barracore on PacBio Hi-Fi sequencing. We aligned the new chromosome level scrub J reference with Minimap2. called variants jointly per trio with sniffles 2, and built phased parental assemblies with hi-fi ASM. So this is the pipeline that I've been working on, trying to make a reproducible pipeline that can be used on any non-model species. And so standard tools assembled into a non-standard pipeline. There are 7 stages. Stages 1 and 2 are the alignment, then joint calling across the trio. Stage 3 is a genotype filter, so offspring carries the variant, both parents are the reference, with the quality of at least 60 based on previous literature. Then it splits into 2 independent paths in order to maximize filtering with 2 different baselines. So path A throws out anything that is in a tandem repeat, which is the biggest source of false structure variant calls. This was necessary because, as you'll see in a little bit, we got hundreds of structure variant calls, and that should be raising red flags. That's not possible. These methods do unfortunately include a lot of false positives, and so a lot of manual filtering is necessary. So path B independently revalidates every candidate by aligning the offspring's reads against the assembled genomes of both parents, which is a different tool with a different reference. These different filters capture different kinds of error. Only candidates that survived both of these filtering steps went on into manual review, which was done with IGV. So here's the filtering cascade. Here's what that produces stage by stage. There's this raw candidate pool in blue, 1100 to 1450 per trio. A lot, way too much. That's obviously a red flag and is much too high, so there's a need for heavy filtering. After tandem repeat filtering, you lose 77 to 85% right there. So that's really reassuring. That's telling us that a lot of these are just in an area where false positives are very common. The independently validated set, which were 12 to 34 per trio, was a 97% cut. But the number that matters is on the far right in red, candidates passing both of the filters that I just described across all 5 trios combined, there were only 15. Of 1,213 that cleared the repeat filter, only 15 also cleared assembly validation. So the 2 filters catch almost completely non-overlapping sets of false positives, which is the strongest signal that the survivors are real. It's not the same false positive being located in both. There's double filtering for this. So those 15 still needed a human eye. I wanted to make sure that this was what it was saying it was doing. It wasn't just locating even more false positives through these filtering. So I used IGV Viewer to do manual inspection so I could see the parent, offspring, and reference and see if any of the mutations were artifacts. So if something was found in baby that was also found in mom and dad, that's not a true de novo mutation. But if it was found in the offspring and not in the parents, that's a true de novo. So of the 15, 3 I am 100% confident are true de novo. 4 are tentative, they look real, but are ambiguous based on parental reads, which holds me back from full confidence, and 8 were false positives. So that shows there's still a lot of false positives. This pipeline is not foolproof, but the false positives clearly all failed in the same way. 1 to 3 reads in a parent that carried the variant, but the caller had scored that parent as a reference because support fell below the threshold. So this was not a pipeline failure, it's a built-in feature of reference-based calling at moderate depth. It was found that the lower coverage trios threw more of these errors than the deeper trios, which is expected. And also reassuring, the deeper the coverage, the less likely you're going to have these false positives be flagged. So the genomic distribution, the 15 were spread broadly across the genome, not just clustered, but there were some patterns. The top panel here on this slide is candidate density per megabase by chromosome, longest to shortest. The dashed line that you see is the genome-wide mean, which is about 1.82 per megabase. What jumps out immediately is right at the edge, these red chromosomes. which are microchromosomes. Chromosomes 28 through 33 run up to 10.6 per megabase, which is about 5 times the mean, which is not an unexpected outcome. This is what Edwards et al. Saw at the population level in their 2025 study. Microchromosomes carry disproportionate structural variation in standing diversity and are de novo candidates. Seeing it in both places corroborates that this is not a filtering artifact, that this is just how microchromosomes are. So gene context, 4 of the 15, so about a quarter, fell inside gene bodies, but every one of these gene bodies were intronic, none were exonic. And that's important, right? 4 hits in gene bodies, none in exons, and that's what you'd expect from random placement, plus strong purifying selection against exonic SVs, which is exactly the signature of a fitness-relevant mutation class. The phenotypic standout is that the 248 base pair that we can see in GTP-BP3, the intronic insertion in trio 4, helps modify mitochondrial tRNAs, and the insertion sits just 130 base pairs from the nearest exon, close enough to affect splicing. This would be a natural candidate for a future study for RNA-seq follow-up. So the mutation rate. So putting it all together, the de novo SV mutation rate in the Florida scrub jay from 3 validated and 4 tentative events across 5 offspring is between 1.66 and 4.16 times 10 to the minus 10th per base pair per generation. So in planar terms, that's about 0.4 to 0.1 validated de novo SV per offspring as an average mutation rate. And the tentative ones and the real count is likely closer to 1 to 2, which is exactly what the previous prediction was. So our observed count overlaps this prediction, a top-down prediction from neutral theory and a bottom-up empirical count from trio sequencing land on the same order of magnitude. And so that shows real reciprocal validation for this pipeline. So I'd like to briefly give a couple implications for these findings. So evolutionarily, avian genomes pick up de novo SVs at roughly 2% of the SNP rate. Their rarity is a real biological signal and not a detection artifact. There's conservation implications because diversity ties to effective population size and mutation rate. Small population species, like the Florida scrub jay, the profile of many endangered ones, recover structural diversity very slowly due to a bottleneck. Protecting the diversity that's already there beats waiting for mutation to rebuild what drift erased. Then, methodological, this pipeline is portable. Any species with hi-fi trio data and phase parental assemblies can run it. The goal of this was not to produce a one-off, but a reproducible framework. Thank you very much. I would like to acknowledge several people who helped me along the way. My advisor, Professor Scott Edwards, for the question, the access to the data, the trust to build this as an undergraduate, and the mentorship has all been so invaluable. I also want to thank Teresa Pagan and Kelsey Lopez, colleagues from the Edwards Lab, for their feedback throughout, and the long-term team at Archbold for access to these samples, as well as Dr. Qian Qin for guidance on constructing my filtering pipeline, and Professor Heng Li for long call D development and support. Thank you all very much.
Thank you. That was a wonderful talk. You should be very proud and excited to have that be your sort of capstone to your undergrad. So congratulations.
Thank you very much.
Our next speaker is Sankar Subramanian. Sankar, go ahead and get started whenever you're ready.
Yeah, I'm Sankar Subramanian from the University of the Sunshine Coast, Australia. So I'm going to introduce you to a very, very simple software used to compute population genetics summary statistics using large number of SNPs. So as our tradition, I would like to acknowledge the traditional custodians of the land, the Gabi people on which we live, work, and study. I pay my respect to the local indigenous elders, past, present, and emerging. Well, so we all know like we have tons and tons of software to estimate population genetic statistics, and just to give you a quick run out, so using one or few genes, you have popular softwares like Mega, Harlequin, DNASP, GenAlex, or GenPop, and with using large number of like genotypes or SNPs. You have Blink, VCF tools, Angst, PubGenome, and .R. Generally, we can classify these software as either standalone computer applications that you can download and install in your computers, or web server-based online software, or the R scripts. So usually, using these software, you need to install them, and or you have to upload your genome data, and most of them involve series of data processing steps. Could be computationally intensive or time consuming, and also they depend on web traffic, internet, and so on. But importantly, all these software require at least certain level of expertise in computer. So the basic idea of developing this software is, mainly, we wanted to come up with a very simple and user-friendly and target for non-computer experts, and which can produce the estimates, population genetic parameter estimates within a few clicks. So I have come up with this idea of a client-side software. So most of these software available are server-side, where you upload your data front end, and it is processed in the back end, and then it produced back the output. So this is the client-side software. It's written totally using JavaScript. So basically, it reads a file and filters SNPs based on your criteria, and then estimates various diversity and population structure measures. And it is, it's a browser-based software, so it is compatible with all browsers and operating systems. And as I mentioned, it only requires the browser, and you don't have to install. And importantly, it reads file from the local computer. So it's just a HTML file that you download and save in your computer, and everything is read within your local computer, and there's no data processing, and it is very quick, and it reads the data like 1 GB data in seconds to minutes. And as I mentioned, importantly, it does not require any expertise in computer. So our main target users are undergraduate students, especially in classes, teaching genetics, ecology, evolution, and biologists with little or no expertise in computer, and installing and running a computer program. And above all, it's overall for anyone who wants to have a quick look at some of the statistics. You've sequenced a large amount of SNPs, and you have hundreds and thousands of SNPs, and you want to have a quick look within like 10 minutes to see some stats. So that is the main purpose of this software. So earlier we developed a similar one using same JavaScript background, and which is mainly meant to produce a tree using gigabytes of data and millions of SNPs. So this program usually rates the VCF file and then produces like trees, and then UPGMA or neighbor joining tree. in different form, like circular tree or so. And it also produces pairwise distance matrix in mega format so that it can be like copied and used using mega for further analysis, and also produces a Newick tree. So using this program as a backbone, now we developed the same JavaScript-based client-side software to estimate population genetics um, parameters. So this is the overall the first front page. So this is being developed. My main idea of this talk is to introduce you this talk. I mean this software, and this is being developed. And this is the main page. As you can see, it reads the VCF file and a population list of populations, and then it'll filter SNPs based on the missing data, and then does some of the analysis like diversity and measures and population structure. So first, you have to, it's a prerequisite that you enter the proportion of missing information that you have like a cutoff. So if you want to keep only the SNPs with or only the sites with 95% information, then you enter 0.05 and then simply click choose file, which will read the VCF file. So first you click this VCF rate button and then open up upload your file, VCF file, and then use the same, but simply click the other button. And so you have to supply a text file containing the sample names and then the population name. So with these two information, this program is able to generate, measures like these, like the average MAF or heterozygosity, expected, observed, the GMRD, and for theta, Watson theta, or nucleotide diversity, we need to, the user has to enter the size of the genome. And we are in the process of adding more measures. And then the output will be something like this. So you will get all these estimates and then their standard errors. So we use simply Bootstrap to estimate these standard errors. And you can also save this file using this button, so to a text file. And for population structure using, so if you can get estimate like AMOA and pairwise FSTs. So if you say click OK, then you will get the AMOA estimates here, and it also lists the proportion of variation explained within and between populations, and the pairwise FST with significance. And so right now we allow something like 1,000 genomes. But the number of SNPs, it depends on your memory. So if you have a large memory, you can input more SNPs. So typically, if the computer is has a 16 GB memory, and it can process up to 2 million SNPs. So yeah, so right now we are also adding other measures like Hardy-Weinberg and base quality filters, haplotype diversity, and also to include Mandel test with the geographical information is provided, and also produce a tree using the pairwise FSTs, and also working on different ways to improve the processing speed. Right now, it takes about like 5 minutes, 5 to 10 minutes for like 100 genomes and 2 million SNPs. So we want to reduce that. And in the future, we also want to add something like principal component analysis. Um, yeah, so as I said, the main idea was to introduce you to a software, and I would like to thank the University of the Sunshine Coast and Australian Research Council and FRDZ and New South Wales DPI. Thanks for everyone.
Wonderful talk. I, for one, love the idea of a quick look at my data. That's easy
Yeah.
to get. So I thank you for working on this. We have time for questions. If anybody has a question, feel free to raise your hand. You do have one question waiting for you in the Q&A when we're done with any verbal questions, but if anybody has verbal questions, now's your chance. And I'm happy to ask Brian's question, which is not as complicated as the last question we had in the Q&A. So the question is, what techniques have you used for assessing accessibility? Like, for example, does it work for people with low vision?
I haven't thought about it yet. Yeah, that is a good suggestion. Yeah, something to consider in the future.
And to add to Brian's question, one of the reasons he was asking is that that can be important for whether or not you can use it in classes because of accessibility needs. So
Yeah, exactly.
yeah.
That's true. Yeah.
All right. Any other questions? Well, thank you so much for your talk. You do have one more question in the FAQ, I mean in the Q&A, if you want to go ahead and take a look at that. We have about 2 more minutes before our next speaker, and I want to start on time in case people are hopping between sessions. This next is our very last, which is going to close out our conference, and it's Katie Eaton. Katie, do you want to go ahead and get your slides up so that when it's time, you're ready to go?
Yes, I will do that.
And while Katie's doing that, I just want to say thank you to everyone. This has been a really great virtual conference so far. And for those of you that are joining in person next month, I look forward to hearing even more about your research. And Katie, we have about 30 seconds.
Okay, and you can see in here, okay?
I can see in here right now you're just in the like regular PowerPoint mode. I mean, you haven't like started the presentation.
Oh, well, that's weird
It
because
popped
I
up
have.
momentarily and then it disappeared. I don't know why.
Huh.
Yeah, I wonder if you just need to switch whatever screen it's pulling for the share.
I only have one screen.
It comes up and then it disappears again. It's very
Oh,
weird.
that's so weird. How about that?
It did the same thing
No.
again.
Oh my God. Okay, hang on. Please
It's
hold.
totally fine because you're the last speaker of the session and we're right on
Okay,
time.
we're just gonna share my whole computer.
All right.
How about now?
Now I think it is coming up. There we go.
All right.
All right, and you are good to start whenever you're ready.
All right. Great. Yeah, thank you guys so much for closing out the last talk of the virtual conference with me. So I am Katie Eaton. I'm a postdoc at LSU in Morgan Kelly and Brant Faircloth's lab. And I am super excited today to be talking to y'all about some of my postdoc work that I've been doing, which is really focused on understanding the genomic basis of hypoxia tolerance in the eastern oyster. So in case you are not familiar with it, this is the eastern oyster, Crassostrea virginica. In addition to being a tasty, depending on maybe your opinion, a tasty appetizer that you can get at many restaurants. These bivalve mollusks are extremely important members of the ecosystem. They are found throughout the Gulf of Mexico, as well as along the east coast of the United States. And you can see that they serve really important ecosystem functions. As you can see in this little video here, I'll play it again. They're really important in maintaining healthy ecosystems. They filter the water around them. You can see this tank of oysters over here filters the water, and you have this clear, beautiful, nice and clean water, whereas this tank without any oysters in it remains silty and sediment filled. So they're very important for maintaining healthy water ecosystems. They also serve to build habitat structure for other organisms to live in. Oh, and we're playing that again. Okay. And so here's an example of an oyster reef. So you can see this is a really healthy looking oyster reef here. We have lots of physical structure that's being built up by the oysters, and you can see that there's lots of biodiversity here. There are many other organisms living on and around that structure that's provided by our oysters. But like everything else in our world, oysters are increasingly threatened by climate change. And while we often think of sort of temperature increases as kind of the most pertinent climate change stressor, what we're also seeing is an increase in the frequency and intensity of other sorts of stressful events, specifically in our world's oceans. Hypoxia, or low oxygen events, are becoming increasingly common in our oceans. And this is really exacerbated by sort of the global warming that we're seeing, where warming ocean waters can hold on to less oxygen. And so it's increasing this frequency and intensity of hypoxia, or again, acute low oxygen conditions in our oceans. And so what can happen under conditions of hypoxia is this beautiful, vibrant oyster reef will start to die and eventually turn into something like this, where we have still that structure kind of remaining from those oyster shells, but you can see that there's a real loss of biodiversity there with the loss of the ecosystem function that the oysters are providing. You can see it's far less diverse, and it's really kind of blanketed by this macroalgae. So obviously this is super concerning, right? But so that kind of leads me into the study that I'm going to talk about today. Luckily, we have some populations of oysters that naturally vary in their tolerance to hypoxia events. And this study in particular is going to focus on two different populations of oysters. We're going to focus on a population from Laguna Madre, Texas. This is our tolerant population. And then we're going to focus on a population from Calcasieu Lake, Louisiana. This is our sensitive population. And we know from previous work from other members of the Kelly Lab that these populations vary really strongly in their tolerance to hypoxia. So oysters from Laguna Madre, Texas can survive for up to like 7 to 10 days under hypoxic conditions, whereas oysters from Calcasieu Lake, Louisiana tend to die off much earlier. And so this really interesting sort of phenotypic variation that we see among these two populations presents an interesting opportunity for us to try to understand the genomic basis of that variation. So that's where I come in here. As I just said, this study is really focused on understanding the mechanistic basis of this variation in hypoxia tolerance that we see between these two populations of eastern oyster. So to address this question, we did low coverage whole genome sequencing for 20 oysters from each of those two populations. And then we kind of took it through just sort of the basic bioinformatic pipeline. We did some read mapping, we calculated genotype likelihoods using angst, and then we did a bunch of downstream population genetic analyses. So just to start off, kind of just looking at neutral population structure, what we see here is that oysters from Texas and Louisiana show really strong genetic differentiation. And you can see this on the PCA here, where we've got our oysters from Texas. Over here, they are extremely separated from our oysters from Louisiana that cluster more towards the left-hand side of PC1. In case you weren't convinced, we also did some admixture analyses, and I have never seen an admixture plot that looks so perfect, where we see really strong and complete separation of our individuals from our two different populations. So yes, there's lots of baseline neutral genetic differentiation between these two populations. We can also look at where in the genome that genomic differentiation is occurring on a Manhattan plot here. So here on the x-axis, we've got our chromosomes of the oyster genome, and on the y-axis, we have FST, which is our measure of genomic differentiation. And what you can see is that there's big time genomic differentiation across all of the chromosomes of the oyster genome. There's not really one specific region of the genome that sticks out as extremely different between our tolerant and our sensitive populations. So what do we do about this? How do we identify which of these highly divergent regions of the genome is involved in generating that difference in hypoxia tolerance phenotype? So as I said, we have all of these major large regions of the genome that are highly differentiated. How do we narrow down which of these regions is involved in hypoxia tolerance? Because we suspect that hypoxia tolerance is going to have an impact on the fitness of these oysters, we're suspecting that this hypoxia tolerance trait is going to be under selection. So we're really interested in identifying adaptive differences between these two populations. And we know that a localized reduction in genetic diversity within a single population is often a signature of recent selection or a selective sweep. So we did some analyses to compare the genetic diversity or the pi between our two different populations, so our sensitive and our tolerant populations. And we identified certain regions of the genome that showed localized reduction in genetic diversity, specifically in our tolerant population. I've just highlighted one of them on the Manhattan plot here, but there were several regions of the genome that kind of stuck out in this analysis. But we're going to zoom in on this one right here towards the end of chromosome 4. This region on chromosome 4 that shows a localized reduction of genetic diversity in our tolerant population contains this gene called RAB1A. RAB is a really diverse family of proteins that has many different functions, but RAB1A in particular has been implicated in the stress response, in reactive oxygen species regulation, and in apoptosis in other studies, which leads us to believe that it's possible that this gene may be somehow involved in the hypoxia response in our tolerant Texas oysters. So just kind of wrapping things up here, we asked what the mechanistic basis of variation in hypoxia tolerance is among our two different oyster populations. We found that there's really major genome-wide differentiation among our two populations across essentially the entire genome. But we did find certain genomic regions that have reduced diversity in Texas, indicating that they're possible recent targets of selection. We also suspect that RAB1A, which is located in that locally or that low diversity region in our Texas population, may be a recent target of selection that's implicated in this hypoxia response. With all that being said, this is still an ongoing project. I'm still working on several more analyses for this, so stay tuned and keep your eyes out for a paper coming out hopefully sometime soon with more information on what's going on between these oyster populations. With that, I would love to just thank my postdoc advisors, especially Morgan Kelly. All this data came from her lab and some students in her lab. But Morgan Kelly and Brant Faircloth, as well as all of their labs, have been super helpful at LSU. And then I want to thank, obviously, the LSU Biological Sciences, as well as the National Science Foundation for funding this work. And with that, I'm happy to take any questions.
Thank you so much, Katie. That was a great talk.
Thank you.
Any questions? Everyone's very shy today.
It's the end of the day. It's Friday night.
That's true.
Yes.
We have one question. You can go ahead and call on people as you want, or I can. Yeah. All right.
Go for it,
You can
Genevieve.
unmute yourself.
Hello. I was curious as to what the average FST was, because looking at your plot, it's all over the place, but what did you
Yeah,
find?
it's quite high. It is about, I want to say like 0.25.
Oh, wow. So do you think
Yeah.
these might be like different subspecies or something?
Yeah, well, oh,
Okay.
the Zoom is recording, so I don't want to say anything that's going to get me in trouble. But we think it's possible, yeah. We're not sure. But they're clearly extremely different.
I have a question to throw in there if no one else is. Has any, do you have any idea of what the effective population sizes would be in the species? 'Cause I forget if oysters are like one of the highest effective population size species or if it's something else that I'm thinking of, but I'm sure it must be huge. And if it is, then that makes the FST results even more surprising.
Yeah, so oysters are really cool. They are so off the top of my head, I don't have like a number for you in terms of effective population size, but they are broadcast spawners, and obviously a marine species. So I would hazard a guess that their effective population size is quite high. Um, I'm an oyster transplant. I did my PhD on fish, so I'm actually like new to the oyster system. So I'm a bad person to be asking this question to.
Still an interesting answer.
There's a question in the Q&A too.
I see that. Okay. From looking at the variation, can you guess when the selective sweep may have happened? Yeah, that's something that I want to look into. I have not looked into that yet, but hopefully stay tuned. Upcoming.
Awesome.
All
Well,
right.
that brings us to our time. Thank you so much to all of the speakers and to our wonderful tech help person, Riley. And I hope to see you all who will be there in person in a few weeks. Thank you. Wonderful conference. Great job, everyone.